Okay, thank you. My name is Kedumetse Mogwera. I'm a research scientist in NADI under the parasitology section. I'll be presenting passive surveillance data on ticks and tick-borne pathogens. I'll start by giving a brief background on our institution for those who are not familiar with it. NADI was established in 2015 by merging NAVTEC, BNVL, and Department of Agricultural Research in order to increase agricultural research. So that was the main mandate. In BNVL, our main focus was mainly on diagnosis. Research was lagging. So through the measure, it is intended that we will also improve on agricultural research and contribute. So the mandate of BNVL is diagnosis and surveillance and research on animal diseases, and to ensure food safety and quality control on meat and dairy products. And we also for monitoring residues of veterinary drugs and pesticides for public health. And now to our main topic. As already discussed, tick-borne diseases are a constraint to livestock production. The economic and social impact is felt especially in rural poor farmers who depend on livestock for livelihood. Ticks are vectors of disease-causing protozoa, bacteria, viruses. The prevalence depends on the climatic conditions. So the tick-borne diseases that we are going to focus on are heartwater, theileriosis, babesiosis, and anaplasmosis. These are the ones that we diagnose in our lab. So heartwater is caused by Ehrlichia ruminantium and transmitted by ticks of the genus Amblyom. It is a notifiable disease, and the signs can be mild up to acute. It may even lead to death. And then anaplasmosis, it's a rickettsial parasite. The causative agent is Anaplasma marginale in cattle and Anaplasma ovis in small ruminants. The vector there is Rhipicephalus species. And the associated symptoms are anemia, fever, weight loss, abortion, and death. And then babesiosis, the causative agent is protozoa of the genus Babesia, and the transmitting vector is the Hyaloma ticks, also characterized by fever, anemia, anorexia, and death. Then theileriosis, caused by Theileria species, with Theileria parva causing high mortality. It is also transmitted by Rhipicephalus species. Some of the symptoms are fever, swelling of the lymph nodes. And through the study, we are going to demonstrate the trends and spatial distribution of ticks and tick-borne disease from 2017 to 2024 from the samples that were submitted to the lab. We rely only on data on the samples that were submitted. We did not go out and do any survey. So through this, we can establish baseline data for further epidemiological studies and guide or give a guideline on prevention and control strategies, looking at the trends that we'll discuss. So how did we get the data? We extracted the data from our laboratory management system from the samples that were submitted for testing. The samples were either blood or tissue smears, and we used microscopic examination, GIMSA staining, the gold standard method. That's the only test that we did. So here is just a demonstration of how we arrived at a positive test. The top picture there is GIMSA stained Babesia species inside bovine erythrocytes. The small arrow there, it shows the Babesia species inside is a positive, while the big arrow is a negative. That's how we diagnose. And then here are the results. The trends between 2017 and 2024. Hot water is the most prevalent across all the years. It's the one that's a major problem. Then in 2020, there was an outbreak of filariasis between 2020 and 2022. The outbreaks there occur sporadically. So the only major concern is hot water. Also in 2020, there was an increase in anaplasmosis cases. And then the disease by species, caprine and ovine are the most affected by hot water, and then anaplasma affected only bovine species. For Babesia, the canine, Babesia canis, it was in dogs. So Babesia was only diagnosed in dogs across the surveillance period. And then this is the spatial distribution of the tick-borne diseases. So we used the positive samples to map where the samples originated. So it occurs mostly in the eastern part of the country where the climate is suitable for ticks, the warm and humid climate. Also, the hot water hotspots, the Muchudi, Gaborone, and Molepolle, we have realized that we receive a lot of positive cases from those areas. They are the ones that are most affected by hot water. And this is the tick distribution. So I guess it corresponds to the tick-borne disease occurrence. Now, the limitations of the data, it does not give a true prevalence and distribution because it only depends on the samples that were submitted. There could be under-reporting, so it does not give us a true prevalence and distribution. So there is need to do a study where we can surveillance, where we can collect samples, ticks and blood, and do pathogen diagnostics to determine the prevalence and the distribution. Also, the data, there's a lot of missing information through the species. Some records do not have the species, the ages, which could also help improve the study. And then our future plans in the section, we intend to introduce a multiplex PCR assays for hematozoa, also to develop an antemortem qPCR assay for heart water. This one is ongoing. Currently, we only rely on the microscopic. So we are developing a quantitative PCR assay, which will also enable us to characterize the strains that are circulating. So that we can, the characterization will also assist us in determining whether the vaccine that is being used, whether it's suitable. So we also intend to do tick and TB pathogen surveillance and further epidemiological studies with our partners, BUST and DVS, through the epidemiology unit. Thank you. That's the end of the